bioinformatics is looking at the code of life and trying to understand how to read and write it. So, to introduce ourselves, I am Krystal Thomas White. I am actually not a hacker. I'm a biologist. I am a PhD candidate at Loyola University in the Department of Microbiology and Immunology. And I study the bacteria that live in and on you, the human microbiome. So in particular, I like bacteria, the little things. This is my husband, Patrick Thomas. He, in fact, is in this industry. He has a background in software development, but he is currently working as a pen tester at NeoHapsys here in Chicago.
All right, how's the volume there? All right. Okay, so first thing we want to do is why this talk? Why are we up here giving a talk? Why are we bringing a biologist to a hacker conference? This really comes out of Krystal and I sitting down, having dinner regularly together and saying, how was your day? How was your day? Talking about the sorts of stuff that we're doing and realizing night after night after night that we're talking about some of the same things using different words. And that just started to blow me away, and seeing those connections emerge, not just casually, not just the terms that we use, but the actual ideas really got fascinating. So we started putting some of those together, and it eventually sort of accreted into a talk. The other bit is that if the last time you checked into what biology is doing, what life science is doing, if the last time you were aware of that was kind of high school or college, what is possible now is going to really blow you away. The sorts of practices, the sorts of things that are going on in lab to look inside of living systems and figure out how they tick is truly incredible. We're going to talk about some of that. Hopefully I give you at least a taste of that today. The trajectory of these things is coming together, and the sorts of things that we're good at are the sorts of things that are beginning to be done in the lab. And then productivity of scientists, of individual labs, the sorts of things that might take an entire team of people 10, even 5 years ago. Now, advances in technology, advances in miniaturization, those sorts of things are meaning that individual grad students have access to massive amounts of data that used to have 10 or 15 heads on it. And so now what they really have is a data problem.
And we need help with that data problem, I can tell you. There we go. So as biologists, we're really good at finding stories and finding patterns and looking at data and interpreting what that means for the biological system. Our issue is that right now the amount of data we're getting out of these machines and these one-time run experiments is so massive that you can't do what my PI does, which is paste it all on a wall and take a look at it. You need to have ways to manage that information. And we were never trained to deal with that amount of data. We're learning completely by the seat of our pants, and we're not doing very well. I can tell you for a fact that very few biologists have any idea what the command line is. The terminal to them is the screen of death. They just freak out when they see it. But it's really important that we start having that type of teaching and that type of dialogue so that your expertise can be translated to help our field. So the types of things you guys do every day can help us immensely. Linux, shell, scripting, files and file types. Just changing between file types can be a big issue. And the other thing I'm going to say is that most biologists live and die by an Excel spreadsheet. And all of you are probably going to laugh at that, but that is so true. And it's something we do need to get out of. But again, no one's really told them or told us the better way to do it. So the question for you is what's possible? And what we're trying to explain to you now is it's not huge problems that we need your help in. It's really small ones, things that are second nature to you. An afternoon of your time writing a short script can change my life immensely. And by changing my life, you might actually just improve your life or your health or, you know, fruit flies and squid. But it's a process. So there are 3 parts to our talk. We're going to make sure that we're all on the same page, first of all. So we're going to give a little basic biology lesson. Don't run out of the room, please. And then we're going to talk about real implications between the two fields. These are not the types of things that, oh, it's just kind of cool, we'll talk about it at a cocktail party. These are actually very deep connections between our two industries. And then we'll get into more what you can do.
All right, so here's the dummy slide. This is if the last time like me you checked in on biology was mid high school or something like that. Kind of, you probably got this still stuck in your head and maybe not much else. All living cells have DNA. That much we know. That's kind of cultural at this point. We know that DNA gets read and turned into RNA. Maybe know a little bit about the process, but not much. RNA produces proteins. Proteins are what plants crave. But that's probably about all we know about those. And proteins do useful things in cells. So this is kind of cultural. This is maybe a reasonable baseline to draw from. If this is where your head's at in terms of that biology is all about just the cells and growing things in agar and stuff, it has taken a huge leap in the last 5, 10, 15, 20 years.
And if this is what you know, then bravo, you know the basics of molecular dogma. But we're going to go a little bit more in depth. You probably also know that DNA is a double helix. Means there's 2 strands. There are 4 base pairs, A's, T's, C's, and G's. And each base pair will bind with its corresponding pair on the other strand. He is right. DNA is read into RNA, which is a single strand, and it's unstable. It doesn't stick around for very long. But when it is around, it does allow for the coding of proteins. In order to do that, the A's, T's, C's, and G's are turned into groups of 3. This is called a codon. Each codon then codes for 1 amino acid. You string a bunch of amino acids together, you get a protein, and the proteins do useful stuff in cells. But the main idea is that the proteins are the machines of your cell. They get the work done. Without this, you wouldn't have cells, you wouldn't have energy, you wouldn't have anything. The other interesting thing is that each codon, even though it's a group of 3, multiple combinations of 3 can code for the same amino acid. That allows for degeneracy in the system. This is a biological system that gets a lot of mutations, radiation, stresses on it, and it needs to be able to adapt. So it has degeneracy built in. And finally, the proteins are very widely in length. They can be very small and they can be very large. The thing that makes them readable is that we know that there is a start codon and a stop codon. And we know what those look like. We know how to find them.
Okay, this looks like life science. Let's make it look like software. DNA is a base 4 representation. A, T, C, G. Those are your 1s and your 0s. The byte width is 3 bits. Remember those codons that we talked about? So we have bits and bytes. Nothing surprising there. They're just different sizes. Here's something bizarre. It has a 1-bit error correcting redundancy. Every single instruction has given over 1 bit, over half of its expressibility, to redundancy. That's pretty much insane. If you're looking at any software systems, to use that much redundancy gives you an idea of how much stress, radiation, chemicals, that kind of stuff, these molecular machines are operating in 100% of the time. We're also looking at a variable width instruction set. We were talking about amino acids produce proteins. So we're looking at x86 or x64, 1, 2, 3 bits, or 1, 2, 3 bytes per instruction. Here we are 10 up to hundreds or even thousands. Massive range of types of instructions that you can have, but we can still enumerate those instructions and know what all of them are. Perhaps at this point this sounds a little bit more familiar. This sounds a little bit more like code that we can look at and reason about biological systems. This looks like code that we can reverse, that we can decompile, that we can begin thinking about what the cells do. All right. If that's true, we should be able to look at DNA, complicated parts of it, and understand something really interesting about the cells.
So I'm going to take you on, or I'm going to tell you a story or take you on a scientific journey, okay? And I want you to come with me. I want you to actually answer my questions, all you guys in the back, okay? I want you to work with me here. There are implications, so let's look at some of those. Scientific exploration starts with an observation. And some bioinformaticians were looking at bacterial DNA, and they were trying to understand why so much of it was repetitive. Why are there repeats all the time? And they ran across this particular pattern, not just in one type of bacteria, but in a whole wide range of bacteria. And it was short, repeated segments, very short, like 20 to 30 base pairs in length, that were spaced by a seemingly random string of other amino acids. So it went repeat, spacer, repeat, spacer. And they saw this everywhere. And they're like, why would you have that? And no matter how much they looked at those repeats, they couldn't find any useful information out of them. So somebody got the idea, let's look at the spacers. So they took those spacers out and they compared them to a database. And lo and behold, they are short segments of viral DNA. This is a bacterial cell that has encoded in its own DNA a regularly spaced out viral genome. Repeat virus, repeat virus. So my question to you is, why would a bacteria contain an array of segments of viral DNA? What was that? Propagate itself?
To propagate the virus?
To propagate the virus. There are examples of that, but that's not what I'm talking about. But that's very good. Want to cozy? What'd you say? Yes. Virus signature. I can't throw that far. That's as far as it goes. Sorry. Thank you. It's a virus signature. Oops. The big question came up with the hypothesis that what if this is an antiviral system, a natural antiviral system? What if those snippets of viral DNA are part of a bacterial immune system that allow the bacteria to identify something invading and destroy it? Does that sound familiar to you guys? Yeah? No? Okay. The question would be how to test it. If you had a system like this, don't worry about the biology, just the code. What would you do to try and test this? Yes, introduce live viruses, but what would you need to do to the bacterial DNA? Monitor it, yeah? You're very close. Yes, alter the match. So you need to insert this snippet of viral DNA into the array and then infect with that same virus. And the hypothesis would be that if the bacteria has that snippet, then it would be protected from the infection. And sure enough, that's exactly what happens. So the other bit is they had to do the biology stuff of expressing all the genes, making sure all the molecular machines are working. But really the key point was that they inserted a known spacer into that array. So they wrote to this code, and then they infected with the virus and protected the cell.
So this is called CRISPR-Cas. You don't necessarily need to remember that. You don't need to remember that, but that's the name of this system. This is the bacterial immune system. This is the bacterial adaptive immune system. It learns from things that it has encountered. It extracts patterns from viruses that it finds, builds them into its DNA, and remembers that. Sophisticated, self-regulated, pattern matching system. It's evolved, not created, and it's phenomenally effective. Lots of bacteria out there. They're doing a pretty good job. It actually works. And then here is where it gets into our territory. This is a file format encoded in DNA and used in living things. We know how to read and write this file format. It's basically a CSV file, except those little repeats, these segments that you use to delimit the DNA, they are amino acids, not ASCII. And that blows me away. We were talking about bioinformatics as the ability to read code and reason about living things. It gets even cooler than just the fact that we can stick a virus in there. If we look at those little spaces of DNA, we can look back in time and see what viruses this bacteria has encountered by taking out those sections of viruses, running them against a database, and seeing what this bacteria needed to be immune to survive. So I ask you this. Is this the only one of these? Is this the only file format, the only data structure that is encoded into DNA? Absolutely not. There's tons of these things out there. And we are the kind of people who are experts in looking at these sort of data systems.
So let's come up with another example.
Reverse engineering 101. Anybody done a little bit of reverse engineering, decompiled something at some point, done a little hack me or whatever? Okay. Suppose, again, set aside the biology for a second. Suppose you've been given a piece of code. You don't know what it does. How do you figure out what it does? And then if you think you've got an idea of what a piece of code does, how do you verify that? I heard run it. Yes. Step through it, run it, any of that kind of stuff. Breakpoints on things. Another way, the IDA Pro way. I hear disassemble, is that what I heard up there? All right. Run it or static analysis, and you can also potentially modify the code. You can comment out pieces, any of that kind of stuff. Same techniques apply here. In biology, if we're going to talk about breakpoints, or if we're going to talk about commenting out pieces of code, knopping out runs of something, like if you got DRM or something, you want to say, oh, let's not check for CD. You can just knock that out and see if you got it right. Biologists do exactly the same thing. They call it tag it and break it.
So we're going to start with break it. In microbiology, we knock out genes. We completely take them out of the bacteria. In larger animals, you can do that as well. You can either turn down a gene or turn off a gene. So yeah, we knock them out. We break it. And then we look at the system and see what happens. So the only difference is that our system is something that's alive. So here's an example. Let's say you had a bacteria that caused a disease, and you think you know which gene is responsible for most of the symptoms. What you can do is go into that organism, take out that gene, and then infect, say, a mouse model or something like that, and see if the symptoms are reduced. If so, you just didn't prove, but you've got a lot of, gone a long way to proving the fact that that one gene was responsible for that disease. Okay? Another example is something that happens in fruit flies. A lot of developmental biology is in fruit flies, and we learn how an embryo develops. So a scientist said, I think I know which gene is involved in the development of wings. So he knocked out that gene, grew up the fruit fly, and sure enough, it had no wings. He just showed, by breaking it, the function of the gene.
Somebody else over there, step through the code. Here we go. Step through the code, or you could do something like a code hit counter. How often does this piece get used?
In biology, we call that tagging it. We usually use something like a fluorescence, so we'll add something that glows to the end of the protein or the end of the gene, so that when it is in the cell, you can visually see it under a microscope. It makes very pretty pictures, first of all, but it's more than that. It's functional. You can see where certain things interact, where they co-localize, where they overlap, and then you can go in and break it and see if they no longer overlap or no longer co-localize. So this is a very powerful tool, not just to visualize and make pretty pictures of cells, but to really get down to how they're functioning.
We're going to talk a little bit about specifically how to get involved in this, but this one in the bottom right is an example of a project that I was working on with someone in Krystal's lab, where this is a human cell, and it's being attacked by a virus. The virus is in either red or green, I actually forget. And there are proteins involved in the cell's immune response in the other color, the either red or green. And so you can tell how effectively the cell is responding to the virus based on when the virus and when the protein are in the same place. Introduce different medicines, see if that gets better over time. And a scientist was dealing with gigabytes and gigabytes of these images, trying to figure out how to go through it. And it wasn't the visual processing part. There was actually a machine that spit out the X and Y of all the greens and the reds all he was looking at was, I've got this file of X's and Y's and this other file of X's and Y's, and when do those occupy the same space? Like, how many lines of Python is that? Like 5 lines of Python? To save a guy hours and hours and hours a week of researching real viruses, real diseases that are affecting real people. Python. Love it.
And we will get into this a little bit more, but that is what we're trying to demonstrate, in that it comes down to the fact that my field has not been trained in the types of skills that you possess, and it could help us a lot.
Then the other thing, and this is just new and up and coming, of metagenomics. This is looking at not just one bacteria, one sample, but everything in some ecosystem. That ecosystem could be soil, it could be water, it could be your stomach, all the microbes that exist in there, or the lining of your brain, or any of this, where you have hundreds of bacteria there. And you can't just focus on one, so you need to develop the sorts of tools that will query across hundreds of files or hundreds of databases. And scientists are used to dealing with maybe one at a time. So now they need to scale this kind of stuff up. I will drink, but it does say big data out there, and that's the sort of problems that they're dealing with.
And if you're at all interested, this is the type of stuff that I do. I study big data sets like this, and he has been a huge help. But if you have any questions about how metagenomics works, or the type of data that comes out of it, or what we can understand based on it, you just come and ask me after.
Couple of Bsides. This is an analogy I've been thinking about for a while. Virus is to sell, as shell code is to process, and whichever side of the house you're on, hopefully the other side maybe makes a little bit more sense. I think it's pretty powerful. This is a very interesting paper. Essentially, some guys coded the names of the paper authors in ASCII, encoded the ASCII in DNA, stuck that into a bacteria, and then started growing it so that if anybody ever sequences that bacteria, they get the authors' names out in ASCII. Apparently, this is something we do for fun now. Genetic DRM, this is getting way out there, but the idea of if somebody has a breakthrough, that they have some life form that does something really incredible, and you don't want people to copy it, life tries to copy itself. Is somebody going to make something harder to sequence so they don't know how it operates? Can you stick sections of code in that make it so that some DNA can still go on growing, but is unsequenceable by normal machines? Somebody's working on this. Realistically, it's probably Monsanto or someone. Someone on our side of the house, somebody on the open source side, should have some idea of what's going on here. And then I'll skip this one for now because it gets detailed, but broken up payloads in gene therapy, really cool. Come talk to me.
Okay, so we've been mentioning this the whole way through. Really what it comes down to is, why are we here? We're trying to not only interest you, but kind of ask for your help. So what now? What can you do? And there really is, I guess, 3 flavors. You can do small, medium, and large. And small is maybe an afternoon of your time, and you can give me or Patrick an email after this. We can give you an email of a scientist that would love some help, or I can hook you up one-on-one and talk and sit down with someone. If you're just interested, we'll give you some actual other websites you can go to. And then medium is more the multiple afternoons, or maybe a weekend, or kind of having like office hours to really help out a scientist. And big pictures, if you're like, I love this, let me really dive into it, we'll talk after, but that requires a beer.
So we're kind of looking at the small, medium stuff right here, right now. As we said earlier, it's kind of helping people get to know what's possible. I can rename 500 files with one command. That's possible? Yes, it is, and it can save somebody a lot of time. Just helping people understand the file formats, reading a man page. It's like when you were learning Linux, if you weren't some savant, it was really challenging to get through all that stuff and understand the underlying concepts. These are people who got through undergrad, got through most of grad school, and then had to learn Linux and don't have any support. So just kind of knowing what's possible, translating for them, having a shoulder to cry on. These are all very useful. And then I want to call out a few different organizations here in particular. Anybody familiar with software carpentry already? Okay. Anybody involved in software carpentry? Okay. definitely check them out. See them on Twitter, follow what they're doing. This is an organization specifically dedicated to this kind of thing, of being a resource for scientists and people doing research to help them use technology well. It's a technology literacy group. They desperately need mentors, people who know their way around command line, Linux, Python, R, who understand databases, any of that kind of stuff, to be in the room and to be a resource during 2 or 3 day workshops. Software Carpentry is doing phenomenal work. They're all over Chicago. They're all over Midwest in the US. So if this is the kind of thing you want to do and you can give away a day or an afternoon or something like that, Software Carpentry, great. Mozilla Science Foundation doing a lot of the same stuff. I don't know if they're specifically active in Chicago, but another group doing a lot of this. Locally focused groups. Anybody from University of Madison or Michigan, Ann Arbor? Anybody? Okay, cool. They have 2 local kind of grassroots groups doing that sort of stuff, and that's another thing to look into. Come talk to me. And then this is brand new this last week. Somebody, as we were prepping for the talk, sent it to me, and it looks like it's trying to be a GitHub, GitHub Stack Overflow kind of mashup focused on biologists and bioinformatics. And they're trying to get some expertise, some people to just check in from time to time and say, oh, I know the answer to that question. So I think it's kind of alpha beta right now, but could be really worth checking out.
And that actually reminds me of 2 things. One is GitHub. I guarantee not a single biologist has any idea what that means. Yet we are asked at many a time to,
my butt
every
is
time you
my butt is changing slides. There we go.
We're asked many a time to basically, you need to do this, just get it done. And we don't know to go to a place like Stack Overflow and GitHub because we've never heard of it before. So we end up trying to either do it by hand, because that's how we know what to do, or to write a code because I've never written a code, but I'm going to try because I was told to, and it's going to be terrible. And that's what all of the stuff ends up being in grad school. It's really unfortunate. And then I had another thought, but I'll think of it again later.
So as we were saying earlier, get your hands dirty. We're talking like 5, 10, maybe 50 lines of code to solve some of the kinds of problems that are costing scientists hours and hours a week, or any of that stuff. Like we were working on a project. Krystal and I were. Well, here, I'll let you go. This is cool. Go ahead.
About a year ago, again, it was one of those things where my mentor came up to me and said, "Find this unique but slightly variable section of code and see how many times it happens in all of these organisms." And I went, okay. And then Patrick said, "Oh, that's easy, regex." And I said, "What?" I've used that term around my department, and everybody gives the same answer. They have no idea what regular expressions are. They don't. But it's so great. And as soon as I learned that, light bulbs went off in my head about all the different types of things I can do, and I've gone and done all these types of things, and it really opened a lot of doors.
This is cool. Anybody ever, just for shits and giggles, gotten DNA and done regexes for, say, like this cancer mutation or something like that? This is weird.
I know. Why would you, right? Yeah.
This is weird. DNA is just strings of text. We have really powerful text processing tools, and they work on stuff that they weren't intended to work on, like DNA. So you can write a regex for a cancer mutation. Is that useful by itself? Not really, but the fact that you can do it is really cool. And then when you start looking at other problems, all of these other tools that you're used to using come flooding back to you. So that's really the point, the interesting part here. We talked about the virus colocalization project earlier. Simple stuff, not high-level models and statistical garbage and blah, blah, blah. No, file management.
And again, the Excel thing, everyone that I know would we are very meticulous people. We wouldn't be in science if we weren't. So we are okay doing something repetitive many, many times. And it takes a lot for us to say, "Oh, I'm tired of doing this." And even when we say that, we'll probably keep going. So for someone to come and tell me, not only can you do it faster, but you can do it more reliably. You can give it to someone that's fairly untrained and they can do it. And we go, "Oh, really? That leaves so much time for all these other things that I've been planning." So it's quite powerful. And again, we're not talking about modifying any sort of existing programs. There's a lot of really wonderful machines out there. It's really what comes out at the end and helping scientists manage the endpoint and what to do with that data. And also, it's not just computational. Is there anyone here that likes to hack more on the hardware side? Yeah? When I was working at UCSF, I had a technician in our lab, did all our computer stuff, and he wrote this amazing program. So we keep all of our samples, all of our precious samples collected from all sorts of things in these massive freezers. So if the freezer goes down, our lives are ruined. Years of work are lost. And this happens, unfortunately, fairly frequently. So he said, that's an easy fix. I'll just set up a temperature sensor to a computer that will just text anyone, even if it's at 2 in the morning, send them an email and a text that the freezer is going out and they can come and save it. Oh my God. How long would that take you? Not long. I had no idea it was possible. And it saved our freezer once and saved us a lot of worry throughout just our day-to-day lives.
A little bit of high-level stuff as we wrap up here. Lots of scientists doing their best to learn the technologies that they need to get their job done, but in so many cases they're learning a single software package or a single piece of software and not the larger skills. We've been through this, we've seen this happen. We don't want to let a generation of really smart people just learn COBOL or just learn Ruby on Rails or something like that, because then you don't learn general computing, you learn a tool. We can save them from that. Science is having this really weird identity crisis right now where it's been the most open thing of sharing ideas and competing ideas and publishing results and having other people challenge it. But the open source idea is really weird for science, of playing with all of your cards on the table, of releasing your source, of releasing your data, of letting other people build on it. That's somehow been a bridge too far for science. Just now, just 2014, some journals are requiring people to submit code and data so that the results can be replicated so that other people can modify those programs and build on it. And as some of these journals have the code published, there are scientists who go, oh my gosh, we could do something so amazing with this, but they don't know how to use Git. They don't know how to get a hold of the code. And so again, we get back to, help me learn what is possible, what to do with this. Just people to be resources for them to ask those kind of questions will help a lot.
And to just elaborate that on just a little bit, what he means by the open source idea, the whole point of science in the academia right now is that you publish or perish. I'm sure you've heard that before. And if you were to give your data away, someone could scoop you. Someone could publish before you. And that's where the fear is. It's not that we don't want our information shared with the world. It's that we want it shared with the world when we're ready. So that's the dichotomy. So I think we actually flew through this presentation. Sorry about that. But are there any questions? We would really love. Oh, fantastic. That's a great question. And
let me repeat the question. He said, have you guys talked to astronomers? And I assume the question comes in two parts of, have you talked to astronomers for help in this? And have you talked to astronomers to see how you can help? So
I would say I don't have any connections in the astronomy world, so my answer is no. And in general, what I see, there isn't all that much conversation. There isn't even all that much conversation between biology and chemistry, let alone physics, let alone astronomy. There should be. I really do agree, but we don't have those connections. What connection I do have is to you all.
I suggest marrying a scientist. It works out pretty well.
Other questions? Yeah.
Krystal, how do you address, you talked about a little bit of the difference between open source mentality, which is put everything out there. You've got professors that are having to
He was asking more about the open source. Sorry, the open source question. How do you deal with that dichotomy that the professors aren't willing to give away their data, but you do need to have this sharing? Is that pretty much right? Okay. I would say that we need to start smaller. We need to start with baby steps. And part of that is that you can think of the code as a tool. So they're not so concerned about distributing tools around. There are papers that publish methods, but in general, the tool gets you to the answer of the hypothesis. It isn't a hypothesis in and of itself. So if you can say to whoever you're working with, look, we're going to have this tool, and all I need is an example data set. I don't need to know what the actual samples are from, or I just need the numbers. You can make them numbers, 1 through 500,000. I don't care. And we're just going to use that as an example to show how the tool works, and we're going to put that online. That would be totally fine. And I think that's the baby step we need to take. And then once people are used to that type of sharing, and they see the types of things that can come out of that, the types of collaborations, then it'll be a much easier conversation.
What kind of issue do you see with that type of collaboration between open source computing environment and academia? How does that work out from your perspective?
So basically the same question of how do we get academia to embrace the open source idea and start putting stuff out? And similar to Krystal, it's the subsets of data and sample data and anonymization and things like that. Some of the same problems that if you want to release a QA database or something like that, and you want to have prod volume, prod complexity data, you have some anonymization procedure, and we've been getting that right for a while, or wrong for a while. But so some of that. But then also, I think there is a mental shift. Who's released something to the public under an open source license? Anything at all? Few hands. Okay. So here was I've done a couple of things, not much, but here was the mental shift that I personally had to get over of, I'm really bad at coding. It turns out. Like, I can do some stuff, but it's ugly. And I thought that people would call me on it. I thought that I'd get like pull requests of, your code sucks. And like, that's it. And I didn't. I got some people saying, hey, I used your code to do this and that, and I got like, it fell over in this edge case and did terrible things, but I liked it, thanks. And so for scientists, it's such an adversarial process. And, you got somebody ready to scoop you, somebody ready to jump on you, somebody who wants to make their reputation on telling you that you were wrong. And to say a little bit that in software, you can air your dirty laundry as long as your dirty laundry is useful to someone, and that's okay. I think that's a mental shift for everybody who gets ready to do software.
But do you feel like, from a computer science perspective, that you're forgiven more easily for messing stuff up? Your boldness, because in academia, it's not necessarily true, right? Because like, if you screw up, you put out the wrong pieces, you put out the wrong data.
If it's actually published, yeah, which is why we're so rigorous before we do the publication. We want to make sure it's right. So yeah.
So.
Yeah,
I think he was asking about is software more forgiving than academic papers and stuff, and I think yes, because they're saying truth is what's in a paper. They're saying this is truth. We're saying it might be useful. My license is the WTF license.
So how would that help, Krystal, from your side in academia? How would that help if that mentality bled over to academia? If it was less about you have to be right 100% of the time, or it's our ass in the line?
Well, I think it's the publication, we do need to make sure that, as he says, it is truth or as close as we can get. However, I feel like the conversation needs to be less restricted. That's kind of what we're talking about in general, that everyone's so isolated. And there's all of these buzzword talks about interdisciplinary this and that, when really that's not what's going on. You have people that rarely talk to the lab next door, and sometimes to a lab across the country, but only at conferences. It's not a daily dialogue, and that really needs to be the part that changes.
Do you find that most refreshing about being in Patrick's world is that you can bullshit each other a little bit and get away with it?
Oh, there's a lot refreshing about this world. Yeah.
What do you find most refreshing from a biology point of view coming into a computer science point of view? How are those environments different that are liberating or helpful in academia? Your perception.
The ability or the willingness to fail. That's always nice to see and to feel. A lot of people are scared about that, but you guys you break something on purpose to see how it works, and that is your mentality. And I do feel a kinship to that because I do that as well, just in slightly more a different system, really. But yeah, that's probably the best part is everyone's willing to talk about what they broke and how they fixed it, or if they fixed it. Thank you. Is there any other questions? Yeah. I have worked with IPython. We mentioned software carpentry earlier. Again, I want to plug them. They're amazing, and they teach IPython in their classes. That's how I learned about it. And it works as a lab notebook format, which scientists are very used to, and allows you to have your code, and then you can comment it on the side, and you can run it step by step. And that works really great for us because we can see it in a format that makes sense. No, it's a really great program. I definitely use it. And yeah, Software Carpentry is a fantastic organization. Please check them out.
Other questions? Thoughts? Beers? Thank you, everyone.
Thanks.